Every living thing on Earth, from bacteria in deep ocean vents to forests, fungi, whales, and human beings, depends on chemistry. Cells copy information, build structures, harvest energy, and separate themselves from the outside world. But at some point in Earth's distant past, there were no cells, no genes, no enzymes, and no organisms at all. There was only a young planet, water, minerals, gases, heat, impacts, lightning, and countless chemical reactions. Abiogenesis is the scientific attempt to understand how that non-living chemistry could gradually cross the threshold into life. It does not describe one magical spark. Modern research treats the origin of life as a sequence of steps, simple molecules forming, combining, organizing, replicating, and eventually becoming systems capable of evolution. The early Earth was dramatically different from the planet we know today. It formed about 4.54 billion years ago, and for a long time its surface was shaped by volcanic activity, asteroid impacts, intense heat, and a changing atmosphere. Oceans eventually formed, creating environments where chemistry could continue for long periods instead of vanishing instantly into space. The earliest evidence for life appears surprisingly early in geological terms, around 3.8 billion years ago, with possible traces even in ancient hydrothermal deposits. That short window matters. It suggests that once Earth became habitable, the processes leading toward life may not have required impossible luck, but could have emerged naturally under the right chemical and physical conditions. Life today is built mainly from four families of molecules. Lipids form membranes, creating boundaries around cells. Carbohydrates, including sugars, store energy and provide chemical building blocks. Amino acids make proteins, which perform much of the work inside organisms. Nucleic acids, especially DNA and RNA, store and transmit hereditary information. A successful theory of abiogenesis has to explain more than the appearance of one molecule. It has to explain how these categories could arise, interact, concentrate, and support one another. The challenge is not simply making organic compounds. The deeper challenge is showing how scattered chemistry became organized enough to persist, copy patterns, use energy, and improve through natural selection. One famous clue came from the 1952 Miller-Urey experiment. Scientists simulated some proposed early Earth conditions by using water, gases, sparks, and heat. The result was striking. Amino acids, the building blocks of proteins, formed from simpler inorganic substances. The experiment did not create life, and scientists still debate how closely its setup matched the real early atmosphere. But its importance remains enormous. It showed that biologically important molecules can emerge through ordinary chemistry. Later discoveries strengthened that idea. Amino acids and other organic compounds have been found in meteorites, comets, asteroids, and even star-forming regions. The ingredients of life are not unique to Earth's surface, they appear to be part of cosmic chemistry itself. Once basic organic molecules exist, the next question is how they become more than a chemical soup. Molecules need places to gather, react, and avoid being diluted. Minerals may have played a major role by providing surfaces where molecules could stick, align, and catalyze reactions. Cycles of wetting and drying near ponds or volcanic landscapes could have concentrated ingredients and encouraged longer chains to form. In other models, pores in rocks or deep-sea vents acted like natural laboratories, trapping chemicals in tiny compartments. Concentration is essential because life depends on networks of reactions. A molecule floating alone is just a molecule. A molecule repeatedly interacting with others in a structured environment can become part of a system. Many origin of life researchers focus on the idea of an RNA world. RNA is fascinating because it can store information, like DNA, but some RNA molecules can also behave like catalysts, helping reactions occur. That dual role makes RNA a possible bridge between chemistry and biology. If early RNA-like molecules could copy themselves, even imperfectly, they would introduce heredity. Imperfect copying means variation. Variation means some versions could become more stable, faster copying, or better at using nearby resources. That is the beginning of evolution. The RNA world hypothesis does not claim that modern RNA simply appeared fully formed. It suggests that RNA, or something similar before it, may have been central in the first self-replicating systems. But RNA is not the only possibility. Some scientists favor metabolism-first hypotheses. Instead of starting with a molecule that copies information, these ideas begin with energy and reaction networks. On the early Earth, certain minerals could have catalyzed chains of chemical reactions, producing increasingly complex compounds. If a network helped regenerate its own ingredients, it could become autocatalytic, a system that reinforces itself. This would not be alive in the modern sense, but it could be a step toward life. Metabolism-first models are attractive because all living cells depend on energy flow. Before genetic instructions became sophisticated, there may have been chemical cycles that captured energy, built useful molecules, and created the conditions for later replication. Another crucial step is the appearance of boundaries. Life is not just chemistry, it is chemistry contained. Cell membranes separate inside from outside, allowing useful molecules to stay together and harmful conditions to be partly controlled. Lipids are important because some can spontaneously self-assemble in water into bubbles or vesicles. These simple compartments could trap RNA-like molecules, amino acids, or metabolic networks. Once reactions happen inside a boundary, selection can act on whole protocells. A compartment that grows, divides, or protects its contents better has an advantage. Membranes may have started leaky and primitive, but even imperfect boundaries would have changed the game by turning loose chemistry into organized chemical individuals. Deep-sea hydrothermal vents offer one of the most compelling settings for abiogenesis. In particular, alkaline white smoker vents create natural chemical gradients, with differences in acidity and charged particles across mineral barriers. Modern cells use similar gradients, especially proton gradients, to generate energy. That similarity is intriguing. Some researchers suggest that early life may have borrowed from natural gradients before cells evolved their own internal machinery. Vent structures also contain tiny pores, mineral catalysts, heat, and a steady supply of chemicals from Earth's interior. In this view, the first lifelike systems may have formed in rock compartments before evolving membranes that allowed them to leave the vent environment and become independent cells. Other origin scenarios place life closer to Earth's surface, in shallow pools, volcanic fields, beaches, or even within the continental crust. Surface environments offer sunlight, wet-dry cycles, and chemical concentration. Subsurface environments offer protection from impacts and radiation, plus mineral-rich water flowing through rock. No single location has been proven. The real answer may involve several environments contributing different stages. Organic molecules could form in one place, concentrate in another, and assemble into more complex systems somewhere else. Abiogenesis research often works this way, not as a search for one perfect birthplace, but as an attempt to reconstruct a plausible pathway through many overlapping early Earth settings. Scientists also study LUCA, the last universal common ancestor of all modern life, to infer what came before. LUCA was not the first life. It lived millions of years after life began, and it was already complex compared with any original protocell. Still, because all known organisms descend from it, LUCA gives clues about ancient biology. Shared genes suggest it used DNA, ribosomes, the genetic code, and energy systems related to chemiosmosis. It was likely anaerobic, meaning it did not depend on oxygen, and may have used pathways suited to mineral-rich environments. LUCA is like a fossil written in genomes. It cannot show the first step, but it reveals what early life had already achieved. Abiogenesis has never been observed from start to finish in a laboratory, and scientists do not pretend that the problem is solved. Instead, they test pieces of the puzzle: how amino acids form, how nucleotides might assemble, how membranes self-organize, how catalysts arise, and how chemical networks sustain themselves. Each result narrows the gap between non-life and life. The most important lesson is that life's origin may not require a violation of nature. It may require ordinary chemistry under extraordinary conditions, repeated over immense time, in environments that naturally favor complexity. The boundary between non-living and living may have been crossed gradually, through stages that were not yet alive but were no longer simple chemistry. The search for abiogenesis also shapes the search for life beyond Earth. If life emerged quickly once Earth became habitable, then similar processes might occur elsewhere, on planets with oceans, moons with subsurface water, or worlds with active geology and chemical energy. Astrobiologists look for carbon chemistry, liquid water, energy sources, and environments where molecules can concentrate and react. We know only one living planet, so caution is essential. But abiogenesis turns a profound mystery into a scientific question. How did matter become biology? The answer may reveal not only where we came from, but whether life is a rare accident or a common outcome of a universe rich in chemistry. If you found this helpful, please like this video and subscribe for more explainers.